This video will include images of human remains. This is for educational purposes. If you're interested in this content without the inclusion of images of human remains, I am considering a podcast option. I just need some time to record episodes and to find somewhere for those episodes to live. Also, apologies if I get any of the pronunciations incorrect in this one. In 1755, midwife Sabina Orlich died in Vác, Hungary, at the age of 62. She was buried in the crypt of the town's Dominican church. Preparing a body for burial in this crypt involved the deceased being dressed in clothing and placing them in a pine wood coffin. These coffins were painted with the name of the deceased, the years in which they lived and died, and images representing the occupations and jobs that they had during life, like a miner's pick and shovel. Other images painted onto these coffins include crucifixes, skulls, bones, and memento mori inscriptions. The record created by these coffins is how we know so much about Sabina Orlich and the other occupants of the crypt. The use of these coffins, along with the microclimate created inside the crypt, had an unintended consequence. These different variables work together to create possibly one of the largest mummy collections in the world. The environment inside the crypt was generally cold and dry, and the pine wood coffins protected the bodies from any humidity that might be in that crypt. Wood chips inside the coffins absorbed moisture from the bodies. Essentially, the bodies didn't putrefy. They dried out and became naturally mummified. 265 people were buried in this crypt until 1838, when the entrance to the crypt was blocked up and closed. When the crypt was rediscovered during restoration work on the church in 1994, the builders discovered the well-preserved remains of the crypt's occupants. Of those 265 people buried in the crypt, approximately 70% of them are partially or completely mummified. Although 2 individuals showed evidence of medical dissections in the forms of cuts on their chests and abdomens, none of the individuals buried in the crypt showed any evidence of artificial mummification or embalming. In addition to the crypt, the church also had an ossuary containing the remains of approximately 40 individuals. An ossuary can be a chest, box, or a building that contains bones. While some of these remains were also partially mummified, the ossuary doesn't have the same kind of data available for its occupants as the crypt and the people in the crypt's coffins. On their own, the coffins are incredible works of art, and it's equally amazing that they've been preserved for so long. But the combination of the archive of information from those coffins and the well-preserved human remains within them makes this mummy collection honestly sound too good to be true. When we think about mummified individuals for whom we also have their names and a lot of detail about their lives, it's easy to think about Egyptian pharaohs. The difference here is that ancient Egyptian pharaohs were mummified on purpose and constructed their tombs to preserve their remains and all the grave goods that they would need in the afterlife. These preparations and the treatment of their bodies, it was all intentional, because pharaohs had the money and resources to do all of that. The people buried in the crypt in Vác weren't god kings, they were ordinary people. This crypt was a burial site for middle-class families, and that's another factor that makes this collection so incredible. The fact that these individuals were rediscovered in 1994 is also really important. There's a lot we can learn from mummies and different methods of data collection and analysis that aren't really always possible with human remains that are completely skeletonized. X-rays and CT scans are great, non-invasive ways of studying mummies, but when the Vác Mummy Collection was found, DNA and ancient DNA analysis was becoming more common, and not just for human DNA. A year before the renovation of the church and the reopening in the crypt, Spigelman and Lemma published the first study of detecting the DNA of an infectious disease in ancient human remains. And they did this with a disease the residents of Vác would have been very familiar with during the time that the crypt was being used: tuberculosis. Tuberculosis is one of a handful of infectious diseases that can infect human bone and change their morphology, what they look like. Most skeletal changes will happen in the spine, and the general paleopathology of tuberculosis is a big topic for another time. But most cases of tuberculosis won't actually impact the skeleton and leave behind any evidence that we can see in bones. Tuberculosis can infect multiple parts of the body depending on where the bacteria travels to inside the human body. But the most common clinical manifestation is pulmonary tuberculosis, so an infection in the lungs. Skeletons won't still have lungs, but a mummy, especially a natural mummy, might have their internal organs preserved. And that's part of why mummies became really popular for studying tuberculosis during the 2000s. So how would we get started studying diseases in mummies within the Vác Mummy Collection? Step 1 is radiographs, which is the fancy word for x-rays. One of the first studies of the Vác Mummy Collection started with x-rays of 168 of the crypt's 265 individuals. These x-rays showed calcified lesions in the lungs and other indicators of tuberculosis, including changes in the spine. But these are only potential indicators of tuberculosis. They could be evidence of infections from other diseases. So, if the pathological changes we can observe in human remains can't tell us exactly what caused them on their own, we can use DNA to look for the pathogens that cause the diseases associated with those lesions. The 168 individuals in this study provided a total of 350 samples for DNA analysis, taken from the lungs, the pleura, the layer of tissue lining the thorax and surrounding the lungs, abdomens, hair, ribs, teeth, and clothing. The idea of using 350 samples and all of that lab work is exhausting. It makes me glad that I only have about 23 samples and all of my controls. A lot of DNA studies of human remains from the past and the archaeological record don't have sample sizes as big as the one in this study. When the scientists sequenced the 350 samples, they used what's called targeted sequencing to look for DNA that is unique to the bacterium that causes tuberculosis. There are multiple bacteria that can cause tuberculosis in humans. That's a story for another time. They targeted an insertion sequence, which is a segment of DNA, called IS6110. We're not terribly creative with our gene names. IS6110 is used in most studies of tuberculosis DNA, and the results of this study were pretty impressive. 55% of the 168 individuals tested positive for the presence of tuberculosis DNA. 27 of these individuals had both tuberculosis DNA in their samples and x-rays showing pathological changes in their remains. So, what does this mean? What does this tell us about these people and their lives? The presence of tuberculosis DNA in their samples means that, at the times of their deaths, these people had tuberculosis infections. It doesn't necessarily mean that they died from tuberculosis, as they might have had comorbidities or died from something else when they also had tuberculosis. A lot of tuberculosis infections are latent, meaning the bacteria is inside a person's body, but it's not making them sick yet. They don't have any symptoms and can't spread the disease to other people. But if they become immunocompromised, it can turn into an active infection. During the time that the crypt in Vác was being used, tuberculosis was becoming much more widespread. The spread of this disease went hand in hand with the Industrial Revolution in Europe. Populations were growing and becoming more dense. There were a lot of people living close to each other, and that's a good environment for a crowd disease like tuberculosis. At the same time, malnutrition and poor sanitation made it a lot easier for people to contract infections. The fact that we have the names of most of the occupants of the crypt means that we can find family members, like father and son Joseph and Anton Negrovitz. This means that we can study the relationship between kinship and infectious disease. Joseph died in 1793, and Antal died in 1803, only 10 years later. CT scans of their remains didn't reveal any evidence of the pathological changes associated with tuberculosis, but DNA samples from their ribs and fibulae, leg bones, tested positive for IS6110. A similar study looked at a woman and her 2 daughters, who all died between the years 1793 and 1797. Samples from all 3 individuals tested positive for tuberculosis DNA, and the mother was actually infected with a different strain of tuberculosis than both of her daughters. So, the scientists found 2 distinct strains of Mycobacterium tuberculosis. While we can't know exactly what caused the deaths of these people, we know they were all infected with tuberculosis. Tuberculosis spreads from person to person through airborne respiratory droplets, so it makes sense that it would spread among family members if they were living close to each other or within the same household. Tuberculosis isn't the only disease that can cause changes in the human skeleton, and it's not the only infectious disease that was widespread in Europe during this time period. When midwife Sabina Orlik died in 1755 at the age of 62, her skeleton showed changes that could suggest tuberculosis or syphilis. 3 of her right ribs showed remodeling on their visceral surfaces, so the sides of the ribs that face the internal organs, the viscera. Lesions or remodeling on that part of the ribs are suggestive of a chronic pulmonary infection. So, scientists got samples from Sabina's ribs and sequenced DNA from them. They didn't detect any DNA belonging to the bacteria Treponema pallidum, the causative agent of syphilis. But they did find a mixed infection of 2 different strains of tuberculosis. Those studies are all pretty great, but tuberculosis isn't the only disease that has been explored using DNA samples from the Vác Mummy Collection. In a study of 51 samples from 20 individuals, scientists found one individual with an interesting gene mutation. In order to rule out potential contamination of the samples, the researchers made sure they didn't have the same mutation. Okay, I'm gonna apologize. I'm probably gonna butcher this pronunciation. Adenomatous polyposis coli, or APC, is a tumor suppressor gene. A tumor suppressor gene helps control how cells grow. So, mutations in tumor suppressor genes can lead to uncontrolled cell growth, and then to tumors and cancer. One of those 20 individuals in this study had the E1317Q missense mutation in their APC gene, a mutation known to predispose people to colorectal cancer. A precursor to colorectal cancer is the formation of adenomatous polyps in the lining of the colon. Most of these polyps are benign. They're harmless, and they can be removed during a colonoscopy. However, they can develop into cancer if they're not removed. Why look for something like APC gene mutations in mummies? Cancer isn't a new disease. Studying cancer and genes associated with cancer in past populations allows us to study past cancer epidemiology and evolution. We can explore questions of when different mutations in tumor suppressor genes developed and how cancer impacted people in the past. The study of cancer in past populations can be referred to as paleooncology, because we really like to slap the prefix paleo onto a lot of things within paleopathology. So we've covered paleooncology and general paleopathology with the Vác Mummy Collection. So what else is possible? Paleoparasitology. The preservation of soft tissues makes mummies a good source of information about parasite infections in past populations. Antal Simon was a priest, teacher, and director of the Institute of the Deaf from 1802 until his death in 1808 at the age of 36. His remains didn't show any evidence of lesions or injuries, nothing to hint at a possible cause for his death. An autopsy, CT scans, and samples taken for histological analysis revealed a worm, several centimeters long, and worm eggs. After analyzing the shape, size, and structure of the worm and the size of the eggs, the authors of this study concluded that the most likely species the worm belonged to is Ascaris lumbricoides. Ascaris lumbricoides is a parasitic roundworm that causes ascariasis. I probably didn't say that properly, but it's one of the most common worm infections in the world. This has been a lot of different studies, all based on one population, one collection of individuals. The Vác Mummy Collection isn't the only collection of crypt mummies in the world, and it certainly isn't the only group of mummies that has taught us about disease in the past. But to sum all of this up, let's revisit that study of Sabina Orlik. That study was published relatively recently, at least at the time that I wrote this script, which shows that the Vác Mummy Collection is still important for paleopathological research. The mummies are now housed within the Department of Anthropology at the Hungarian Natural History Museum, but some of the collection and some of those painted coffins can be visited at the Tragor Ignác Museum in Vác, next to the church where they had originally been buried. I have seen this referred to as the Memento Mori Museum online, so that, I don't know if that's easier to look up. Please let me know what other mummies you would like to learn about, and thank you for watching.